In this video, we're going to talk about cell division. So we're going to multiply cells by dividing them. We're going to look at how prokaryotic cells, bacteria, how they replicate, and we're going to look at eukaryotic cells like ours and how they replicate. In this diagram, you can see a very simple bacterial cell. Bacterial cells have one circular chromosome, their DNA molecule, and sometimes they have this little extra piece of DNA called a plasmid. We're not going to worry about this at this point in time, but in other videos we will talk about plasmids. They can carry genes for antibiotic resistance. We're going to focus primarily on this chromosome, and we're going to look at how these bacterial cells will replicate. When bacteria replicate, there's basically two steps. In the first step, the DNA has to replicate. And in the second step, the cell has to replicate, and that is called binary fission. You can see in this simple bacterial cell that we have one DNA molecule, and it's a circular chromosome. There is one origin of replication, and this is where the DNA is going to start to replicate. This process of DNA replication is a semi-conservative replication process, and that means we're going to separate these two strands of DNA, and each strand will act like a template for a new strand of DNA. So the new strand of DNA is shown in this diagram in red, and each new strand is being made from an old strand that acts like the template. From the origin of replication, DNA replication will happen in both directions, adding new nucleotides until we have two copies of that DNA molecule. So remember from previous videos, all the guanines are going to combine with cytosines, all the adenines will combine with thymines. This is exactly the same process in every living organism. When we have these two new replicated DNA molecules, now we can basically split the cytoplasm. The cell will elongate, and then it will pinch in the middle and basically just divide into two new cells. It's important that the DNA replicates before the cell divides because each new cell needs to have a copy of the DNA molecule. So these two new daughter cells are exactly the same as the original cell. So in this example, bacteria are reproducing through a process that is asexual. They are the same. The original cell is exactly the same as the two new daughter cells. How do bacteria have genetic variation? Genetic variation is a very important concept for all living organisms. If populations of organisms don't have genetic variation, then one single change in the environment can kill off a whole huge portion of the population. When we have genetic variation in organisms, then different kinds of environmental changes, some of those organisms are going to survive. So how do bacteria acquire new DNA if their replication process is an asexual process? They acquire new DNA in three different ways. One way is to copy the DNA. In this example, it's showing the plasmid is going to replicate itself and move over into the other cell. This process is called conjugation. When bacteria share DNA directly from one cell to another cell, that is conjugation. Sometimes bacteria can acquire new DNA from viruses. This is a bacteriophage virus that can infect bacterial cells, and this process is called transduction. And the last way is there can be DNA fragments just surrounding the outside of the cell. Picture your throat. inside your throat, you have all kinds of bacteria, normal bacteria organisms that live there and help protect you from pathogenic bacteria. And sometimes you might have an overpopulation of, say, strep bacteria, and you get strep throat infection. Different bacterial organisms can exchange DNA. So that conjugation process that we talked about first, that can happen between a streptococcus and a staphylococcus. They don't have to be the same species. Now, let's suppose some of the bacterial cells in your throat die. That DNA from those cells now becomes part of that environment. Other bacterial cells can take up that DNA. So this process of picking up DNA from the environment is called transformation. It's kind of cool that bacteria can pick up new DNA from its environment, and then it can kick out old DNA. So bacteria actually have a very rapid evolutionary process because they have many different ways of acquiring genetic variation. So that's how bacteria replicate. It's the simplest type of cell division. Actually, cool fun fact is that mitochondria, the organelles in our cell that make ATP, they replicate inside our cells in the exact same way. They have DNA, they copy it, and then they split, and we make new mitochondria. So now we're going to focus on how eukaryotic cells replicate. Eukaryotic cells are more complex. There's more chromosomes, there's a nucleus. So the process is a little bit different and there's multiple steps. A couple of things that I want to point out before we begin the steps of mitosis. We have two processes in eukaryotic cells. Mitosis, which is what we're going to look at in this video, but we also have meiosis, and this is a slightly different process we'll look at in a different video, and this is how we produce sperm or eggs. So we are focusing on mitosis. This is a cell division mechanism that occurs in somatic cells. And the purpose is to replace old cells or to make new cells when we grow. So where are our somatic cells? Somatic cells are basically any of the cells in our body that have differentiated and become a specific cell type, like your skin cells, your liver cells, your blood cells, or bone cells. So those are somatic cells, whereas germ cells, they are going to be the cells in the testes or the ovaries that can become sperm or eggs. So while we're looking at the mitosis process, we are focusing purely on somatic cells. In this diagram, we can see what we call a karyotype. A karyotype shows all of the chromosomes from one cell. This is a human karyotype, and humans have 46 chromosomes. We get 23 chromosomes from our mom, and we get 23 chromosomes from our dad. When we look at a karyotype, we can see that these chromosomes are all lined up in pairs. These pairs are called homologous chromosomes. So this is chromosome number 1. We get one of them from our mom and one of them from our dad. Chromosome number 2, one from mom, one from dad. All the way up to our sex chromosomes. The sex chromosomes can be either an X or a Y. When you are a female, you have 2 X chromosomes, and when you are a male, you have an X and a Y chromosome. Another interesting thing that you would be able to tell from a karyotype is if you had an abnormal number of chromosomes. People that have Down syndrome have 3 chromosome number 21s. This is just interesting because a lot of people think that the more DNA you have, the more complex the organism is, but that is not necessarily true. Potatoes have more DNA than we do, and so do crabs. Slime mold has more than fruit flies. So the number of chromosomes does not determine the complexity of the organism. I want to explain the difference between a homologous chromosome and a sister chromatid. This is a very important terminology differentiation point so that the steps of mitosis, and later when we talk about meiosis, that those will make sense. So it's really important to understand the difference between these two terms. Homologous chromosomes are the different chromosomes that we get from each parent. So if this was chromosome number 1, we would get one of them from our dad and one of them from our mom. They are both chromosome number 1s, but they can be slightly different. Let's suppose gene number 1 was for hair color. And let's suppose you got a blonde hair gene from your dad and you got a brown hair gene from your mom. Both of these genes are for hair color, but they could be slightly different. These slightly different genes are called alleles. This will be an important topic also in another video when we look at genetic inheritance patterns. When you have different chromosomes coming from each parent, they are called homologous chromosomes. When we are going to have cell division, we have to replicate the DNA. In humans, in all of our cells, if we're going to make new skin cells, we have to copy the DNA so that each new skin cell has a copy of all 46 chromosomes. Just like bacteria, they have to replicate their DNA before they can split their cytoplasm so that new cells have all of the DNA. When we replicate our somatic cells, then the 2 of the chromosomes will be exactly the same. After DNA replicates, we call those chromosomes sister chromatids. Sister chromatids come from DNA replication, and they are exactly the same. So homologous chromosomes can be slightly different, but sister chromatids are exactly the same. After DNA replication occurs, the sister chromatids will stay stuck together at this little center point called the centromere. And then later, during different steps of either mitosis or meiosis, these will separate. Another concept that I want to talk about before we get to the actual steps of mitosis is the whole cell cycle process. There's only about 2% of the time of a life cycle of a cell where it's going through mitosis and dividing. The other 98% of the time, or it could be a little bit different in other cell types, but most of the time, cells are not dividing and they're doing normal cell things. The genes are being transcribed into RNA and they're making proteins like insulin hormones or collagen, and those cells are functioning like normal cells. The amount of time that cells are actually going through mitosis is a very short period of time. So during that stage when they're not dividing, we call that interphase. But interphase has 3 different stages. I want to look at that. In this diagram, this whole portion of time is interphase. Most of the cell cycle is actually going to be here in G1. So G1 is a growth phase where the cell is doing basically normal cell functions. So if it's a liver cell, then it is going to be getting rid of toxins and storing blood sugar and doing normal liver things. When the cell is going to divide, then it will leave this G1 and it will start to go through 2 other phases of interphase. This one is called S phase. S phase is when the DNA is going to replicate. So then we are going to make all of our 46 chromosomes. We are going to copy them. We will have sister chromatids. And then the cell will actually contain 92 chromosomes. Then the cell will move into G2. This is a second growth phase, and this is where we're going to have more preparation for cell division. Things like replicating mitochondria or the centrioles. This will happen in the G2 period before the mitosis phase. Then mitosis is going to occur, and there are basically 4 steps. There's a prophase, a metaphase, an anaphase, and a telophase. The very last step, which is sometimes considered a separate phase from mitosis, is called cytokinesis. And this is like the binary fission part of bacterial replication. This is when the cytoplasm is going to divide. So we start with 1 cell, we go through this whole process, and we end up with 2 cells. After mitosis, these 2 cells are identical. Just like bacterial replication through binary fission, when we make new liver cells or new skin cells, they will become liver cells and skin cells, and they will be exactly the same. Over here, I want to point out that some cells don't ever go through mitosis. And this is called G0, and this means that the cells basically stay arrested in that normal cell functioning stage, and it never replicates its DNA or other organelles or divides. So 2 examples of cells that are really important to know that never divide. Can you think of a couple of examples? One is our neurons. Neurons do not go through cell division, and neither do our skeletal muscle cells. So the muscles that you were born with are the muscles that you have. When you grow bigger muscles, you're making each muscle cell bigger by adding protein filaments to those muscles. You're not making new muscle cells. So neurons and muscle cells do not go through mitosis. Now we're going to look at the actual steps of mitosis. There's a few key things that happen in each of those steps, and I want you to pay attention to what the chromosomes look like, which chromosomes are separating. In this diagram, we can see interphase. During interphase, notice that the chromosomes are all unwound. So this diagram is showing an organism that only has 4 chromosomes. There is a chromosome number 1 from the mom, and a chromosome number 1 from the dad, a chromosome number 2 from the mom, and 2 from the dad. So there are 2 pairs of homologous chromosomes shown in this example. When these chromosomes go through S phase, they will duplicate. So there's actually 2 chromosomes in each of these. So there are sister chromatids that came about from DNA replication in S phase. As we move through the stages of interphase into G2, so interphase, we have a G1, we have an S phase, this is when the DNA replicates, and a G2, when we're going to prepare for cell division. One of the organelles in eukaryotic animal cells that's very important for cell division are the centrioles. The centrioles are going to replicate during the G2 phase as the cell is preparing for cell division. So at the very beginning of cell division, at the beginning of mitosis, we will have 2 pairs of centrioles. All of the DNA chromosomes will be replicated. When you look at a cell under the microscope, the DNA kind of just looks like it's all over the place inside the center of the cell, and there is still a nuclear membrane. The cell still looks basically like a normal cell. Then we are going to enter mitosis, and the first stage of mitosis is called prophase. In prophase, there's a few key things that are going on. Number 1, we can see the DNA is condensed. The DNA is fully condensed, and now you can see separate sister chromatids. So we have, say, chromosome number 1 from mom, chromosome number 1 from dad, and there are sister chromatids for each of these. Chromosome number 2 from mom and 2 from dad with sister chromatids. The sister chromatids are from DNA replication. These are fully condensed in prophase, and in a microscope, you can see condensed chromosomes. The other thing that's going to happen, number 2, the centrioles are going to start moving towards opposite ends of the cell. Sometimes the opposite ends are called poles, like the North Pole and the South Pole. And they will produce spindle fibers. These spindle fibers are made from microtubules. These are very important because they are going to connect with the chromosomes to help move them to opposite ends of the cell. The third thing that happens, the nuclear membrane is going to start breaking down. Nuclear membrane will break down because we can't keep the chromosomes inside of a nucleus and then separate them. And one last thing is the nucleolus. Do you remember how the nucleus has a nucleolus? It's a dense region of the nucleus where we make ribosomal RNA. That no longer is visible under a microscope once mitosis begins. So we say that the nucleolus disappears, it just breaks apart and it will reform again later. In metaphase, we can see that the chromosomes line up at the center of the cell. Sometimes this center is called the equatorial plane. All 4 of the chromosomes are lined up. So 1 from mom, 1 from dad, 2 from mom, and 2 from dad. And the sister chromatids are still connected at the centromere. The centrioles have moved to the opposite ends of the poles, and the microtubules, the spindle fibers, are connected to that centromere region. There is a region called the kinetochore where the spindle fibers are going to bind. And in the cell micrograph image, you can see that the chromosomes are lined up in the center of the cell. The next stage is called anaphase. Anaphase is when the chromosomes start to move to opposite ends of the cell. So which chromosomes are separating? The sister chromatids are separating. They're moving to opposite ends of the cell because these spindle fibers start to shorten. They get broken down, and as they're broken down, they pull the chromosomes to opposite ends of the cell. In this diagram, you can see that the chromosomes are coming apart. Then we have telophase. In telophase, this is distinct because you can see the indentation forming in between, in the center of the cell. These indentations are formed by actin filaments. The other things that happen, the spindle fibers are completely gone. We still have the centrioles here. The new nuclear membrane is starting to reform. The nucleolus will start to reform. It's not shown here, but the little dense region, the nucleolus, will also start to reform. And then the very last step is called cytokinesis, where the cytoplasm separates. So now we have 2 new identical cells. They are exactly the same. We started with cells that had 4 chromosomes, and we are finishing with cells that have 4 chromosomes. A 1 from each parent and a 2 from each parent. So these cells are exactly the same. And now these cells will go into the G1 phase, and they will act like normal cells. So that is the basic process of mitosis. We are replicating somatic cells. We are making identical daughter cells that will replace cells that are old or damaged, or if we're growing. This is just showing a little bit more specifically the process of cytokinesis. During cytokinesis, we are forming actin filaments in the center of the cell. This forms a cleavage furrow, and this will eventually pinch off completely and form the 2 new cells. And lastly, here's a little bit of a summary of some key points. Somatic cells go through mitosis. The purpose is to replace old cells or grow new cells or to repair injuries. If you cut your skin, you have to grow new cells. This is a concept I forgot to mention. When we have 46 chromosomes in a cell. That is called diploid. When we split those homologous chromosomes, then it's called haploid. All of our somatic cells are always diploid. They have 2 copies of each of the chromosomes. This 2N means that you have 2 homologous chromosomes, one from mom and one from dad. Later on in another video, when we look at the production of sperm and eggs will be haploid. They will have 23 chromosomes instead of 46. That way, when a future sperm and egg combine, they form a diploid organism. So somatic cells are diploid, and in humans we have 46 chromosomes.